Okay, hey everybody. Let me just check and make sure this is working, all right? I'm always paranoid that I'm the only one here because I didn't push some button right. Let's see. Yep, okay, so I'm seeing some chats. That looks good. Okay, so somebody in the chat, let me know if you can see me all right. Okay, good, good, good. Alrighty, let me just share this real quick. Okay. So this week we're going to be doing the antibiotic screen. So everybody should have this little file, right? Well, see if we can get this to focus on it. No. Oh, I had it for a second. Oh well, you get it. It says G418 antibiotic 200 milligrams per milliliter. So that's milligrams per. That's same as milligrams per or micrograms per microliter is milligrams per milliliter. So first thing we're going to do is going to be pretty similar to last week. Last week, what everybody did was split their cells using trypsin. How did that go for everybody? Like, is everybody able to get several flasks? You should have a few flasks to work with. I'll show you guys this. So, I think you guys are mostly working with the larger flasks, the 25 centimeters. These guys. I've got some little ones, but that's what I usually use. The 25s are actually nicer. You can get longer growth times out of them. I usually use the smaller ones just because it's sort of more efficient, uses less media over time and stuff. But okay, so let's take a look at some of my cells so you have an idea of what your cells should look like. Okay. All right. Just share the screen. Let's see if I remember how to do this. All right. Everybody see that all right? Okay, so this is a pretty good flask. It's like middle growth, you know. These aren't fully confluent. I've got another flask that's more confluent. This is one I split on the 21st, so it's only a couple days old. And this is what they'll look like once they've reattached and they're healthy and everything looks good. But you'll see like some round cells. That's just cells that aren't happy, and a lot of them you kind of tell aren't attached. See how they're wiggling around a little bit when I give it like a little bit of a wiggle and then stop. They keep moving because they're not really attached. Those are dead cells. If you have lots and lots of those and very few of the attached elongated cells, then your culture is not very happy. You might need to change the media or something like that. Now this one is at a much higher confluency. You can see, I mean, it is packed with cells. There's like no empty space or like very, very little. So this is what they look like when they're like as grown as they're going to get. Any more than this, and they can get really, they can just start dying on you. You can tell between the two of these. Oh, well, let me maximize me here. Okay. You can tell the slight difference in color. Maybe the lighting's okay. So this is the newer one, and it's got a pinker media. And this is the older one. It's got a yellower media. And so you can tell that this one's kind of near the end of its life. So this is the one I'm going to use. So I'm going to use the one that's a little bit older. We're going to trypsinize it down, and we're going to, instead of splitting it into a bunch of flasks, which you can take the opportunity to split it into a flask or two if you want, but we're going to actually split it into a 12-volt plate. So this is your 12-volt plate. It will be like this guy. So you can see I've got media in my wells, and you can also see I've got cells growing in it. So these are treated just like the flasks are. And you may need to actually make a little modification to your microscope. Nope, wrong button. There we go. All right, so it'll have these two little clips that are designed to hold slides. You can just take the screws out, and that'll give you a bigger stage to work with. Because when you put these 12-volt plates on there, they take up the whole thing. Let's see. Can my cells be contaminated if I have COVID? I mean, possibly. I don't know. It depends. I don't know if HEK cells express the right receptors for COVID to be able to successfully do them. They're supposed to be kidney cells. I don't think COVID attacks kidney cells, but these are also highly mutated cells from the 70s, so they're not exactly normal cells either. But you can see I've got a plate with lots of cells growing on it. Plates are a little bit weirder to see because they don't really fit on the microscope very well. And so you may have to constantly adjust the focus as you go from well to well, right? Just because they don't sit perfectly flat. But okay, I can back this up. This is with the slightly higher magnification. If I back out, you can see a little bit broader space in the wells. Yeah, as far as replacement cells, I'm trying to answer some of the questions as I go, but some of these I may have to get to at the end. I'll have like a whole question section there. But as far as replacement cells, a lot of them have been sent out. Don't know how many have arrived yet, but there will be more sent out continuously because everybody's going to kill cells. When I first did this, I killed all my cells repeatedly. I had to get sent probably 3 or 4 different flasks before I was able to keep one alive. Yeah, so all we'll have to do is, if you are behind or if your cells are all dead right now, don't worry, let us know. Send us an email, odin@theodin.com. We'll send you more cells, but we need to get, but everything will be recorded, so you'll have access to everything. So if you fall behind, as soon as you get them, you can just start catching up. But okay, so first thing we're gonna do is clear our space. So just like last week, you want to have a clean workspace. I usually just use alcohol, but you can also use alcohol and wipes and different stuff. You're going to need gloves and you're going to need your mask. That's the biggest thing for avoiding contamination. You know, the biggest source of contamination is always you. Your body's just completely covered in fungal spores, bacteria, archaea, all kinds of different stuff that will try to grow in your media. Now, the media does have antibiotics in it, designed to reduce that as much as possible, but you know, there's always antibiotic resistant stuff all over the place. So, use your gloves. I always spray my gloves down, and I also spray all the way up my forearms. So, I use a ton. I'm gonna go ahead and put this back in the incubator, and this can go back in the incubator. Yeah, so if your cells are growing faster than you want, so this is a good question. If I can get that to pop up. Okay, great. So if your cells are growing too fast and they're overrunning, you use fewer cells when you inoculate. So when you do the initial digest, it depends, right? So it's like the number of cells that you start with really has a big impact on the number of cells or how fast they grow, right? Because they double. So if you start with 100 cells, you know, they'll double in their doubling time to be 200. But if you start with 500 cells, they'll be 1,000 in the same time. So the number of cells that you start with has a huge, huge impact. If they're overrunning you, like if I were to digest this one, right, which has lots of cells per, you know, cubic centimeter, the trypsin solution that I end up with, that I start my next cell with, or my next flask with, is going to have more cells than if there were fewer cells in this flask. So you're starting a denser culture, and also if you use, so you can kind of scale that, right? I usually use between 50 and 200 microliters of cells. Once you finish the digest, you've just got some cells. I use between 50 and 200, depending on what I'm trying to do. If I need lots and lots of cells because I want them to grow super fast, I use like 200. If I need very few because I want them to grow like next week sometime, then I'll use like 50. If I want them in 4 days, 5 days, I'll usually use around 100 ml. So you can count, or 100 microliters. So you'll dial that in with experience, because it depends on a lot of stuff. And yes, the media does have antifungals along with antibiotics, so it should help protect you with both. But okay, so you should have a bottle that looks something like this. Yours may look like this. Don't worry, they're the same thing. So you're going to clear your space. One thing I like to do is put down a paper towel and spray it with alcohol, just so I have somewhere to put things that's not just sanitized, but it's also sanitizing. So that way, if I have to put down a cap or a filter or anything, I have a surface I can put it on that is like continuously being cleaned by the alcohol. So I'll put my mask on. Okay, and you should have like a little bottle, something to pour, discarded fluids into. You should have your media. It's important that the media be pre-warmed. Yours will be in a little bottle, but it's important not to put refrigerate. You store it in the refrigerator, but don't just dump it on your cells, because they won't like it. They don't like cold, and cold shock is bad. So take your media, warm it up. You can either just stick it in your incubator if you've got an incubator, or on your heat mat, give it about half hour, or you can take it and stick it in the microwave, but be really careful not to overheat it. It'll overheat faster than you think. I usually give it like 5 seconds at a time and then check. And even then, it's easy to overheat. So be careful with that. Okay, so let's see. We're going to be using a 12-bowl plate. And if you look in the classroom, there's a description of what we're going to be doing. So you should find this document in the classroom, and it will have this diagram in it. So that diagram is going to tell you which well. You don't have to actually stick exactly to that, but it's a good guideline. It'll help keep you from getting confused. These plates have sort of angled corners, so you can't put the lid on backwards. So you can just label the lid on this one. Okay. So we'll need a 12-bowl plate. We'll need our antibiotics. We'll need our media pre-warmed, and we'll need our trypsin pre-warmed, because you don't want to put that on them cold either. So we're going to take our first flask. So this is going to be the flask. This should either be a flask that's getting old, or you should have a backup flask. Don't digest your only flask because you're gonna lose all your cells if you do. So make sure that you have another flask growing, preferably with a few more days on it, and you have this flask that you're going to digest and use. If you don't have an extra flask, do that first, and make sure you got a couple of flasks at least that you trust before you start this next part. Okay, that's important, or you'll kill all your cells at this stage. You will technically have your control wells that you could try to rescue from, but it's much easier just to have flasks. Okay, so first thing we're going to do is we're going to clean up like that a little bit. Anytime you're working with mammalian cells or human cells, it's important to make sure that everything's as sanitized as possible. You'll never be perfect. That's what the antibiotics are for. But why try to discover how many antibiotic-resistant organisms live in your house? So I like to get everything as open as possible before I dump my media out, because I don't like leaving them dry for any amount of time, because that's stressful. They don't live in the dry. So give it a little bit of a swirl. That way any dead cells will just go with the media. All right, pour that out. Doesn't have to be anything special. Grab it like that. Put it at 1,000. So we're going to use 1 milliliter or 1,000 microliters. This first step is just a wash step. In the previous class, we used PBS to do this. This time we're just using the trypsin. Give it like a little swirl. And pour that out. Change tips. Get another ml. Now we're going to let that digest for a minute. Stick that in the incubator. Trypsin is an enzyme. It's a digestive enzyme. It digests the proteins that help that the cells use to stick to the plastic. So we're just going to digest those away, and we'll end up with cells that are free of that. But okay, we're going to need one of these guys. These are 15 ml tubes. And what we're going to do is we're going to add about 15 ml of our media to this tube. And you can do it by adding media directly to each well and then seeding them with cells manually. The trouble is, okay, so good question here. But it's harder to get things right, and you're more likely to contaminate stuff just because there's more steps in and out of the plate. It really doesn't matter. You can use 1 ml or 2. 1 ml, the biggest thing is that you cover the bottom of the flask. You have 25 centimeter flasks are twice the area, so you can use 2 mLs of trypsin, but honestly, it'll digest about the same. You could use a lot more or a lot less. The biggest thing is the ratio of media at the end to trypsin still needs to be like 3 to 5x. So if you use 1 and you're doing it into 15 mLs of media, that's plenty. If you end up using 2, you end up with a lot of media that's the ratio is different, I guess I should say. So it should still be good enough. I wouldn't go too crazy with it though, or you'll end up in a situation where you don't neutralize the trypsin at the end. And that's an important step because neutralizing the trypsin keeps it from continuing to digest the proteins of your cells, and eventually it'll kill them. So I usually use between 1 and 2, but it depends. So just look at it, add 1 ml. If it's not fully covering the bottom, you can add another. Let's see, perform the wash with PBS trypsin solution. So the benefit of performing the wash, so the reason we switched from using PBS instead of trypsin, so obviously PBS is cheaper than trypsin, so that's what most people do. But if you do the wash step with the trypsin, we have run into a situation where basically there's less contamination. So it's very difficult for anything to grow in trypsin solution because it's protease, it digests proteins from stuff. So it's really hard to live in. But PBS, is an okay environment. It doesn't have sugars and stuff, so it's not easy things to grow in, but things can survive in it. So we've had less contamination, plus it simplifies things for the kit and for people to just use the same stuff, because this is way more than you'll use in these experiments. Okay, so we're going to prep this guy. So we're going to add some media. We need to get a syringe and a syringe filter. So what I usually do, make sure my hands are clean before I reach into the bag, because I don't want to reach in and contaminate the rest of them. And you'll see it has one end that's a smooth thing and one end that has sort of a little bit of a screw cap on it. So what you're going to do, The smooth, sort of tapered end is the sort of business end. That's where things go out of. So make sure you're really careful not to touch that to anything. If I touch it to anything, it's the sort of alcohol prep pad I have over here. So you just screw that on there, and it goes pretty easy. But this filter is small enough that, you know, living things can't get through it. All right. So we're just going to filter some media. What I usually like to do, as soon as I open the cap, I put it face down on the alcohol and then cover this with this so that nothing gets in while I'm handling it, pulling the end off, and all that sort of stuff. It can be a little tricky handling all these tubes and stuff all at the same time. It takes a little time to get the hand skills. But okay, that's 10 mLs. I'm just going to filter it just in case my media has been contaminated. Just generally assume anything's been contaminated. Like the media we send out has antibiotics in it, and it's already been syringe filtered more than once. By the time you get it, actually make 14, because we're going to add 1 ml. Okay, if you have any excess, you just put it right back in your yeah. Okay. Then we have this guy. Make sure you cover everything. Make sure you don't pass your hands over stuff. That's a big source of contamination. You've got a tube open with media in it, and you cross your arms over it a bunch of times. Just think of like a rain of bacteria and fungus just falling off of you all the time. Every time you move and there's air movement across your body, it just pulls bacteria and stuff off, and they fall straight down into whatever. So try to be careful that you don't pass your hands over stuff after you've sterilized it. That's a big source of contamination. But okay, so let's check on our cells. It doesn't take long to digest them.
Yeah, they look good.
If you're doing the digest at room temperature, it's a little bit slower. But you see how there's like, I cannot control the focus on this thing. But you know, it's like opaque now, cloudy, and you'll see like what was a sheet of cells on the surface now is kind of like just falling off. And if you look at it under the microscope, it should look like this. See how there's kind of a bunch of isolated individual cells, but also a bunch of sort of flakes. Now these cells, yeah, see the big flakes? We want to break those flakes up. That's just a bunch of cells stuck together, and it'll make sort of a weird knob, and everything will grow better if you can break those flakes up. So we're going to use the pipette to do that. So, let me switch back to me. How long in the warming pad for this? Just check them after about 5 minutes. So I usually digest for about 5 minutes. If I'm doing a bunch of like flasks that I'm prepping or something like that, and I've already started the digest, I'll do it at room temperature just because it's slower. You can just sort of watch it happen. So I mean, even if you do it, if it's too fast for you, you can slow it down by reducing the temperature. If you're just bored waiting on it, you can warm it up and it'll go faster. So okay, so this part, look over, you can see. All right, so I'm gonna pipette them pretty gently. All right, I'm just gonna like pick it up and squirt it back in. And I'll kind of wash the side. Do that again. Wash the side. And that way we break up any of the cells that have, you know, stuck to the side. And looking at them under the microscope, you know, they're much more broken up. All those big flakes are all a bunch of like either really small flakes or individual cells. Okay, so now we're just gonna pick this guy up, add it to this. You can't filter cells into anything because obviously you filter the cells out. That's bad. So now in here is our cells, and they're all nice and diluted. Just shake them around a little bit. This will neutralize the trypsins. so the digest will stop because of the serum in the media. There's like antitrypsin proteins in the serum, and yeah, so you can see that it'll get a little bit of a cloudiness. Maybe you can see it. I don't know. Will it focus? No, it won't focus. Anyway, it'll get a little bit of a cloudiness, and that's your cells. So now, now we move on to our plate. Now you want to label your plate. You don't necessarily have to do this. So there's two ways you can do this. I'm going to actually do the antibiotics today, so I can show you guys. But you may want to give them a little time. I think the protocol in the classroom actually suggests giving them a little time so that they come, you know, they're adherent and they look nice and they look like that other plate that I was showing you guys. But today I'm just going to sieve them and add the antibiotics. I've never tried that way, but I'm confident it would work fine. G418 doesn't kill things quickly usually, so I think they'd have time to adhere and do their normal thing before any kind of problem happens. But okay, clear my workspace a little bit. It's important to have a clean workspace if you want things to stay clean. All right, so hands again. All we're going to do now is seed the plate, and it's important that when you seed the plate, you think about how you're handling it, right? So if you just like flip the lid over and squirt them on everything, there's a lot of room for contamination. So what I usually do is I just lift the lid a little bit and I go under. So the only thing that's ever exposed to the surface between the lid, so that if there's any like, you know, spores up here that are falling, they don't land in my stuff. So the only thing that's ever under there is like the end of my pipette and, you know, the pipette tip itself. So as long as your pipette is clean and your pipette tips are nice and sterile, which they come pre-sterilized, everything should be okay. Now I'm going to use 1 ml, so that's 1,000 on the larger pipette. And you should have some excess media, so if you have some left over, don't worry about it. But okay, so you're just going to fill a well. Each one gets an ml. And it's really doing it like this, especially, you know, since you're not breathing on it and stuff like that, really minimizes the exposure to the air and the environment. It can really help make sure that your cells don't get contaminated. Because contamination is terrible to fight. All right, now once you've done this, your cells are going to be, you know, it's going to be a day before they attach good. And then probably another day before they're sort of happy again. So at that point, you can either, you know, check them and make sure they're growing and look decently confluent, and then you can take those and start the antibiotics. Or if you want to try it like I'm doing it, you can do it here. Okay, so I've got a little bit left over. I'm just going to dump that. Right. If you wanted to, you could start, you know, a flask or something like that. But I know y'all don't have a ton of flasks to throw and stuff. But okay. Now, each well has media and cells. And I'm going to label it. Okay, hold on, there's some questions. This thing's weird about auto scrolling. Who's this guy? Got the wrong channel. Yeah, Josiah is in Josiah's in Ukraine right now, filming with Daria. So I'm filling in for him. Okay. Let's see. All right, so if you look at the classroom thing, it's got 2, 4, 6, 8, 10, so on. That's the concentrations. So if you look at the concentration in the solution, it's got 200 milligrams per milliliter. And you look at this and it says, you know, 200 micrograms, 400 micrograms. Well, 200 milligrams per milliliter is 200 micrograms per microliter, right? That's the benefit of the metric system. So you can take the 200 micrograms per microliter, and that's your first well. So we're going to use our smaller pipette. You should have a pipette that's like 0.5 to 10. And this one's really small. Now, if you let this grow out, they'll look just like your flasks do. You know, you don't want to be fully confluent when you start this. You want, you know, some amount of growth, maybe 50 to 70%, somewhere in there. Okay, so this one's a much smaller pipette, uses the smaller pipette tips, and it allows you a lot of fine control. But since we're going to be using it, we'll clean it up too. So we're going to be using the really tiny pipette tips. These guys, they look like, I mean, they're really small. But what you're going to do for the first one, it's 200 micrograms per microliter. This is 200 micrograms, or excuse me, this is 200 micrograms per microliter. You want to add 200 micrograms to this well, so you're just going to use 1 microliter. The next one is 400 micrograms to the well, so you're going to use 2 microliters. So 300 or 600 is 3 microliters, and so on. So you just keep adding a microliter every time you go to the next well. Oh, see, forgot my mask. I'm trying to communicate clearly. Can you all hear me okay behind this thing? Alrighty. But it's usually better to label things before you start adding things. Helps reduce the amount of confusion. But 1 microliter is kind of tricky to pipette. It doesn't really look like anything, right? Like, I wish I could make this thing focus, but it's a, yeah, you'll see it. It takes a little practice, but it's very, very small quantity. So it can be tricky to even see. But we'll add 200 to that one, and then you'll discard, get a new pipette tip. Then you'll increase it from 1 microliter to 2. Get 2 microliters, which still looks like a teeny tiny amount. Add that to the next one. Same thing, discard the tip, raise it to 3, get a new tip. Three, and so on. So you kind of get the idea. I'll go through all of them if you want me to. I kind of feel like you probably don't. Let's see, are there any questions so far while I'm doing boring stuff? You submerge the tip when adding a small amount, right? You can. I mean, what happens if you're doing like 1 microliter is it'll half the time like run around and just stick to the outside. So it's better if you do, but it also depends on the viscosity of the fluid you're working with. Okay, so we're adding 5, so 1,000, so that one goes there, and then new tip, so on. Let's see. All right, so what's the point of doing the antibiotic selection in the first place? So when you're growing these cells up, so the next step is going to be the plasmid transformation. So we have a plasmid that has, what's the reason we use more in each well than the last? Are we seeking this? Yeah, okay, that's a good question. So the whole point of doing the screen, which I guess I should have explained at the beginning, the whole point of doing the screen is you're trying to figure out how many antibiotics, or like what concentration of antibiotics is going to be needed to kill your cells. And so you would think that that would be like really standard and all the time be the same, but it's not. Unfortunately, it's just sort of a tendency. So because these antibiotics don't just out and out kill cells, you know, there's no like just sort of magic death machine. Everything kills cells by doing something. And so what happens is these cells, or this antibiotic, messes up their ability to make protein. So there we go. So that's all added, and then you're going to label it so that it looks like this, right? So you got, the different concentrations, and you got 3 control wells. So these wells you don't add anything to. Those wells are negative controls. That's the thing against which you're contrasting, your results. And what you'll see is dead cells. So what you should see is, for me, in the conditions that I grow cells, it's usually around 400 to 600 micrograms. And so that's usually enough to kill, micrograms per ml. That's usually enough to kill HK cells in a couple of days. But it depends. HK cells are subject to stress, right? I mean, any cell is subject to stress, but human cells or at least mammal cells are more subject to stress. Obviously, it's much easier to grow bacteria than it is to grow human cells. But if the passage number, so like how old the cells are, the culture of cells, the concentration of things in the media, how cool, like warm or cool it is, how controlled the environment is, how aggressively they've been pipetted recently, all these things sort of add up to stress. And the stress ends up killing cells. So, or at least makes it so that it's harder for them to live. And then you add antibiotics on top of that kill them, and it takes less antibiotics to kill them. If they're living an amazing life and they have no stress, then it might take more antibiotics to kill them. So the reason you do the screening is because your exact growth conditions will be slightly different from mine and slightly different from somebody else's, right? You're growing on a heat mat on your kitchen table and your average house temperature is whatever. I'm growing in an incubator over there. So it's going to be a little bit different. Normally for me, it's between 400 and 600, but it might be 800 or 1200, or it might be 200. So for the next step, the genetic modification step, we're going to be doing antibiotic selection. So right now we're doing antibiotic screen, but we're going to be doing antibiotic selection for the next one. And what that means is we're going to take our DNA, the LAC-Z gene, and we're going to add it to the HEK cells. And that's what allows them to change color at the end of the step, right? It's an enzyme that allows them to change color. So if you're successful, your wells will be different colors at the end. But the DNA that allows them to do that is only given to a few cells. So even the best genetic modification won't modify every cell in the wells. So what you'll have is a mixed population of genetically modified cells and non-genetically modified cells. And the issue is it's easier to grow as a non-genetically modified cell because you're not making extra enzymes that don't do anything. They at least don't help you survive, right? So what happens is when you have a mixed population like that, the genetically modified ones have to do more work. They have to make these extra genes. And so they grow a little more slowly. And so the non-modified ones just can outcompete them. Or even if they're just 50-50, there's just more of them. So there's the larger number of unmodified cells is enough to make them, keep going. So when you do antibiotic selection, what you end up with is a pressure, right? So the cells that are not genetically modified will die, or at least it'll be really hard for them to live and reproduce and do their thing because they're under a lot of stress. The cells that are genetically modified, because the gene that we're adding not only contains, or the DNA that we're adding to them not only contains the LAC-Z gene that helps them change color, it also includes an antibiotic resistance gene for this specific antibiotic, which means those cells will be able to live just fine with the antibiotic. Obviously, there's a threshold. They can't live with infinite amounts, but they can handle a lot more of the antibiotic than the unmodified cells are. So that means it's easier for them to grow and live and do things. So when you do the genetic modification next week, you'll add the amount of antibiotics that you find works good in your conditions this week, and that will allow you to select for the modified cells with, ease. Because if you have a lot of stress in your growth environment, your cells will need less antibiotics to do a good job at selecting. If you have very little stress, your cells need more antibiotics to do a good job at selecting. So we're trying to sort of find that Goldilocks of antibiotic quantity or concentration. So what you're going to do, now that you've got these guys here, if you let them grow out and then add the antibiotics in a couple of days, you can make like a graph. There's an example in the, or I should say like a spreadsheet. There's an example in the classroom, right? It's just under this one. It's on page 2. But it basically is just like on one axis, you know, well 1, 2, 3, 4, 5, 6, 7, and also the wells are labeled, you know, it's hard to see sometimes. It's like 1, 2, 3, and then A, B, C. So you have like A2 or whatever. So, you know, you label your wells, and then you label, you know, day 1, day 2, day 3, day 4. And what you're going to do is you're going to look at them under the microscope. And what you're looking for is dead cells. So you're looking for like little round balls of cells that have detached. And so those cells, you know, are not happy, are not alive. Attached cells are happy living cells. Detached cells are dying cells. So you're going to add the antibiotics at, you know, the various concentrations. Then you're going to measure just by looking at it under the microscope and just take a guess. I mean, it doesn't have to be exact, right? The difference should be significant enough. But what you're going to find is your controls should be happy cells just growing along, doing their thing. And then the 200 should be a little less happy. The 400 should be much less happy. Cells are super stressed out and dying. And then up and up and up, it should get where there's just lots of super stressed out cells that are dying all the time. And what you can do is you're going to record that. So whichever is the lowest amount of antibiotics required to kill all the cells in 2 or 3 days, that's your target. So next week, when you do the genetic modification, you're going to say, okay, all my, you know, 600 microgram cells died in 2 days. So when you do that next week, use 600 micrograms, because that'll do a good job of killing any not genetically modified cells that you have. If your 400s and 200s survived, then it's not enough to select well. If you're you know, if you used a higher concentration, you might kill even your genetically modified cells. So you find the minimum amount of antibiotic needed to successfully kill everything that hasn't been genetically modified. And next week you'll use that to test or to select for the genetically modified ones. Okay, so that's basically what you need to do this week. You don't necessarily have to do it all in one step. You can let them grow out and then do it. But are there any questions? Am I great at estimating confluency? That's a good one. So yeah, it's tricky. The thing though is you don't need, the thing to keep in mind is you don't need it to be accurate, right? So it doesn't matter if you, it doesn't matter. So you're eyeballing it, you know, and like you don't have a cell counter. You know, you could stain them and count the cells, or you could like take a picture on the slide. Let me get it. So the camera software allows you to take photos, or you can just hold it still and count. Okay, let me get my focus right. Okay, so you can take, you know, zoom out into the sort of wider range, so you can see sort of bigger space that makes it more accurate. And then, you know, you look at it and you go, okay, about how much of this space is covered in cells. So for me, I'm gonna say it's about 50%. Then I usually move to another space, like, okay, still about 50, you know, because you can find spaces that are high density and spaces that are low density. So you just kind of like look at several and be like, okay, they're all pretty close to the same, you know, 50-ish, about half. It does not have to be super accurate. If your cells are but the biggest thing is because it doesn't have to be super accurate because you're contrasting it against your controls. So the controls should be dramatically different from the wells that you care about, right? So whatever well they're all dying in, it should look super different from the controls. So it shouldn't be hard to estimate. Even if somebody else or a cell counter would say it's 70 and you think it's 60, it really doesn't matter. Let's see. How much trypsinized cell fluid did you add to the 15 ml? Was it 1 ml? Yes, they used the whole thing. So I added basically all the cells that I digested into that, and then into these wells, which is interesting. The heavy dose wells are already changing color, so they already look like they don't like it. But okay, so when you're doing this experiment, let's see, some things to remember. Make sure that you work really clean. That's the biggest thing, because if you contaminate it, you've only got 4 plates, and you'll need at least 1 for the genetic modification experiment. So try to nail this one the first time and work really clean. That'll give you 3 three plates to work with for the next experiment, which might take a try or 2. Let's see. Any other questions? Oh, something I was going to tell you. Now I'm trying to remember what it was. Make sure you refrigerate your media and stuff like that between, and make sure you change your filters, change your pipette tips. Don't reuse the filters once they get media in them. Just consider them contaminated. Let's see. I'm gonna look back. There was a couple of good questions I skipped. How much trypsin was in the media? 1 ml. Yep. Any benefits from the wash? I answered that one. It's kind of a peripheral question, but yeah. So yeah, humanized mice cells are mouse cells with a couple of human genes added to make them, have some particular trait. Like, say you want to do, coronavirus research, so you need a very specific receptor for that virus to be able to infect, but you want to use mouse cells for whatever reason. Well, you can add that receptor gene to the mouse cells, and then the mouse cells will make it, and then they're humanized. Or you can replace the mouse gene with the human version. And human stem cells are just like a whole different kind of cell. It's like a cell from humans that can grow into many different types of cells. With my cells, okay, so you might get some weird stuff. If your media, so I usually add about 5 mLs of media when I make flasks. If you add a lot less than that, especially if your incubator is dry, it might start drying up. And so like the quantity of liquid will go down. And if it goes down enough, stuff will start to precipitate and crystallize just because there's like not enough water to keep it in solution anymore. So make sure it's not drying up. Usually it'll start to turn yellow or orange once it starts to get sort of a low quantity because it's evaporating too much. And make sure you have like have the lid on tight. They don't need to breathe. The amount of air in the space is plenty of oxygen. They'll run out of space to grow in before they run out of oxygen. Let's see. Yeah, split it. Make sure you have plenty of cells. Okay. Have any idea? Okay, yeah. So I'll go with that quickly. So the first step when they arrive is just take your cells, the first flask, and take your syringe filter and filter a small amount of your media, like 5 mLs, 3 mLs, into another flask. And then take that flask that arrives, you'll get a little flask, and you'll have some of the bigger flasks, right? So use like 5 mLs of media, put them in that, syringe filter directly into the flask, so that flask is ready to receive some cells. And then what you're going to do is just take an inoculation loop, which comes also in the kit, scrape some cells loose, like literally just stick it in there and just rake it back and forth, and it'll just scrape cells off the bottom. It's not as pretty, but it works, and the cells will just be knocked loose and they'll be in solution, and then you can just dump some of that into the other media, and that'll inoculate that flask, right? But just make sure you don't, just dump all of it out. You can actually do it so that you like scrape it and then dump the whole thing, but the flask that you receive will be like liquid full. So dump it down to a reasonable amount, like you can look on the side and there's measurements here. So you can see like this is like graduated. So get it down to like 5. And then you can just, scrape it off, pour some in there. That'll get enough cells going. Let's see. Do you have any idea how to culture ex vivo fibroblasts? So yeah, so this is actually something that I'm working on right now. I've actually got some stuff growing in the incubator. But yeah, so there's a couple of different ways. The main way is take a biopsy, which I've done before. Biopsies suck though. But you take a biopsy, digest the biopsy down, put it in media. The media is a little bit different that you normally use. So you use, and you don't use trypsin to digest it, but use like collagenase, which digests it into like a bunch of individual cells. And then you're going to use like, we've been using 50% F12 and L15 with 20% media, or 20% serum instead of 10% serum, and a couple of growth factors added to it. But then, yeah, you can start a culture from your own body. The other thing you can do is you can actually draw blood, and you can collect, which is something we're about to do soon, is draw blood, collect white blood cells, and then transform them into induced pluripotent stem cells, and then transform them into whatever kind of cells that you want. And then you don't have to do a biopsy. You can just collect, blood cells. So as far as this goes, no, it's not totally necessary. I never do that. As long as your ratio, I generally don't let mine go longer than 10 minutes. That you can definitely over digest them and hurt them. But as far as centrifuging it down or removing them, it's not necessary. If you're growing them in media that has serum, you don't need to do it. The serum will inactivate it. As long as the ratio of media to trypsin is right. If you use way too much trypsin and way too little media, then yeah, it'll continue to work. But otherwise, it's fine. And HK cells aren't very sensitive to it. They've been getting trypsinized since the 70s. The other thing is, that's why we do the wash step. So when you dump off the media and then you wash it with trypsin once, then you dump that off. The whole point of doing that step is to remove any additional media that's still in the thing, or like most of it, because the amount of media that's left can be enough to, make your trypsin not work well. If you just add trypsin and wait, it'd be much slower. So even just a pretty small amount of serum can inactivate the trypsin. So I wouldn't worry about it. If the cell, so technically, I think you can. I've never tried it. I always get new ones. I don't think it hurts the treatment of the flask. If the flask was clean, like you didn't have any contamination that killed them, they just overgrew. So if you get like if your flask overgrows to the point that cells all die, right? Generally, it'll look like really orangey yellow, and the cells will look terrible, and pretty much everything looks dead. But usually there's a few cells, and one thing you can actually do is dump that out, wash it a couple times, and save that culture. So even a really overgrown one can usually be saved, because like half the cells will come off, and then they're not 100% confluent anymore. But usually it's way easier just to get a new one. But if you're running out of flasks, you can. All right. So any other questions? We got a couple more minutes. Let's see. And HK cells are not all that sensitive to stuff. Oh, look, I got options. Oh, weird. So, you know, the biggest thing is just take care of your cells. They're very sensitive compared to bacteria and fungus. Um, try to keep the biggest things are, keep contamination at bay, um, and make sure that your media and everything that you expose them to is a reasonable temperature. So if they've come to room temperature, I usually keep it like slightly warmer than room temperature, but um, you know, if they're just fresh out of the incubator, I try to keep it close to incubator temperature. And because you just don't want to splash them with cold stuff. They don't like that at all. And they also, just keep everything clean. Don't forget to filter, sterilize things. And just be thoughtful and stuff. Make sure that you really understand what you're trying to do before you do it, because you don't want to be like, oh no, I'm in the middle of filtering my cells out before I realize something has happened. So here's a good one. What are the flasks treated with, and where do you order more? So you can order them from us. We have them on the website. So they're not treated with. I mean, I guess they are, but usually they're like plasma treated, or like etched. But basically, it's plastic that's been sort of damaged in such a way that the surface is not so smooth, and like charges and stuff are right, so that the cells proteins can actually attach. Because otherwise, it's actually really hard for cells to attach to plastic. They can attach to glass, okay, but it's actually really hard for them to attach to plastic. A lot of things can't stick to plastic, but yeah, they can treat the plastic so that they can stick with it. Yeah, well, we're working on that one. That one's going to be a little more complicated because we're going to use electroporator and stuff, but we're trying to find ways to simplify it. So that's something I'll be doing a lot of in the next couple of weeks. Okay. All right, so we're closing in on time. Any last questions? Just make sure you label everything. I usually like the fine tip Sharpies. They do a really good job of sticking to the plastic. Like most pens won't write on plastic worth anything. Um, and the broader Sharpies work, but it's just harder to read when you're writing small. So I usually like those. And if you ever need to get, like erase it, it's all alcohol erasable. So just spray it with some alcohol and you can erase it. Okay. So if there's no other questions, next week, just make sure you have us. Okay, one more question. Okay, wonder why slight temperature changes stress out the cells. Does it shock them? Yeah. So, I mean, they're human cells, right? They evolved to live at 98.6 all the time. So, human body is, especially like organ cells, like kidney cells, um, are expecting to be at a very precise, even temperature pretty much all the time. Like the only time your core body temperature fluctuates is when something bad is happening to you, like you're sick and you have a fever or something like that. So your cells do not like to be refrigerator temperature or anything like that, because like that would kill you if your core body temperature reached those kind of temperatures. Your cells don't like it; they're not evolved for it. Um, it damages their proteins and all kinds of stuff. So yeah, so just keep them as close to body temperature as you can. So 37°C. Alrighty. Well, thank you guys for coming. I will start putting some of this stuff away. Make sure you label your stuff. Make sure you work clean. And so by next week, you should have a sense of which antibiotic you're going to use. And it's the one that it's the lowest number that kills all the cells. That's the one, that's your target. Alrighty. Well, everybody have a good Sunday, and I look forward to seeing you guys' results. Make sure y'all take pictures of your cells being blue and stuff, because I really want to see that. I worked hard to make this class interesting, so I'm excited to see the color changes in everybody's cells. All right, y'all have